Hey everyone, in this video section, this is going to be reviewing so that you can get ready for your test. And so just let you know, it covers all of chapter 12 and most of chapter 13. But in chapter 13, the process of protein synthesis, it can be very, very specific. And so I will cover what you should know about the process and kind of stay clear of actually reading the detail part of protein synthesis in your book because I don't want you to get confused. So first of all, you should have at the beginning of this unit reviewed the different scientists involved in the discovery of DNA. So you should have learned about a scientist named Frederick Griffith. He discovered the process called transformation. So basically, he took some harmful bacteria that caused mice to die, and he put them next to bacteria that were not harmful. He put them together and injected them into a mouse, and the mouse ended up dying. So essentially, what he kind of figured was that there was something in the heat killed that somehow got into the harmful strand of bacteria and caused them to become harmful, and then they died. So he discovered this transformation. Now, did he know specifically what transformed bacteria, what went from one bacteria to the other? No, he didn't have that specific name, but he got the idea that there's something inside of us that can move, and specifically in bacteria, and that can change the bacteria. So it's like, what is that? And so then there were these two scientists, Alfred Hershey and Martha Chase, that continued with this process and this kind of idea of something inside of us. And they were studying bacteriophage or bacteriophage. They're viruses that can insert things into bacteria cells. So like, what's this stuff they're inserting? And they determined that it must be DNA. It must be genetic information. And then that's how things are getting passed down. And so another scientist named Oswald Avery was taking this transformation idea from Frederick Griffith and kind of diving into it more and testing different types of macromolecules to determine like, what is this genetic information? And so he determined that it was nucleic acids or DNA. So he determined that, there was some DNA. And so this word DNA, deoxyribonucleic acid, is now kind of a part of scientist terminology. And so now they're trying to find out, well, what does DNA look like? What is the makeup, the molecules of DNA? So then there was this guy named Erwin Chargaff, who kind of looked molecularly at what are the molecules of DNA, and he determined that the nitrogen bases, the percentage of proportion of how many was in each DNA molecule matched. So the adenines equaled the thymines, and the guanine number equaled the cytosine number. So, for example, in humans, he saw that there were 30.9 adenines and 29.4. That's very, 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 very close to being equal. And the same thing with guanines and cytosines. So he determined that adenines bases bond with thymine bases, and guanine bases bond with cytosines. And so we kind of wanted to know what does DNA look like? What's its shape? And they're still trying to figure that out. And so Rosalind Franklin was an amazing scientist who was able to get a picture of DNA through this X-ray process. And she discovered that it was a helical in nature. So that led to the last two scientists, Watson and Crick, to propose the actual real model of DNA in 1953. So let's talk about then this DNA molecule. Now remember, in eukaryotes, DNA is found in the nucleus, and DNA, one of its job is it allows us to make proteins, and proteins is all and everywhere inside of our body, and we need proteins to determine the traits of who you are. So let's look at, let's remind ourselves. So what are the nitrogen bases in DNA? Adenine and thymine, they equal each other, and cytosine and guanine equal each other. They go together. So build the corresponding DNA strand. So if we were to match and make this double helical, what would be the matching base pair? So hopefully you can answer as I answer. G, C, T, A, G. Now, that would make this the double helical strand and complete the DNA sequence for this answer. What are the other parts that make up DNA? Like if you were to think of DNA as a ladder, and like the steps of the ladder are your nitrogen bases, well, what's the side of the ladders? And it would be your phosphate group and your deoxyribose. Ose is a sugar, so yes, DNA has a deoxyribose. A deoxy means without oxygen on its sugar. So again, DNA is made up of repeating nucleotides. So this is a nucleotide, and you need to know what are the monomers that make up the nucleotide. So we have our nitrogen base. So this could be an A or a T or a C or a G. And then we have our deoxyribose sugar. And then we have our phosphate group. And so DNA literally is just repeating this over and over again. So it's one of these and one of these, and it literally just repeats over and over again, over and over again, and then it becomes a helical shape. So it then begins to turn and twist on itself. So it's just DNA is just repeating nucleotides over and over and over again, and then it begins to helical and kind of spiral as it goes. All right, so let's review some pictures and some processes. So you've learned in this unit these three processes: replication, transcription, and translation. So what is this picture? What process is this picture? Which one of these three? Take a minute, look at the picture. And based off this picture, this would be DNA replication. Now, what on this picture is telling you that it's DNA replication? Well, first of all, it's DNA polymerase. ACE is an enzyme. So this is making a copy of DNA. That's why it's called DNA polymerase. And you can notice that it's happening on both sides of the DNA as the helicase is unzipping the DNA. You can see that it's making a new copy here and a new copy here. So this is DNA replication. And so kind of what you need to know about that is what helps make DNA replication. Well, it's this amazing protein right here. DNA polymerase is what really helps make extra copies of DNA. And when do we make an extra copy of DNA? When we're going through S phase in the cell cycle. So reminding ourselves of what's the difference of DNA replication in prokaryotes versus eukaryotes. Well, in eukaryotes, we need many different places for it to start because it's so long. So like we might have multiple DNA polymerases attached different places on DNA because it's so long. We can't just start at one end and then go all the way down. We have to start in many different places because it would take forever for it to finish making a copy. Now in prokaryotes, it's much more simpler because it's a lot smaller and it's a circle. So it literally starts in one spot and it goes in one direction this way and one direction this way until it meets and it makes one easy copy. All right, so in chapter 13, we started to learn about another type of nucleic acid called RNA. So you need to know the differences between DNA versus RNA. So in DNA, here's a picture of the two. You could probably visually see the difference between DNA versus RNA. So first of all, DNA, double-stranded. See, double-stranded. RNA, single-stranded, just half, right? Another thing is the sugar. The sugar is not the same. DNA has deoxyribose sugar, meaning it doesn't have an oxygen on that sugar. And RNA has ribose sugar. So they have two different types of sugars. RNA is much shorter than DNA. And DNA's, one of its nitrogen bases is thymine. And in RNA, thymine isn't there. Instead, it's a nitrogen base called uracil. So now in RNA, there are U's in place of T's. So let's practice a little bit. What is picture number one? What is this? Is it RNA or DNA or both? This is DNA. And you know, because it's double-stranded. And there's T's. You can see thymines. Nitrogen bases in a strand. So what is this? Is this Number 2, is this RNA or DNA? And the answer would be that it's RNA because you see U's, uracil. Contains ribose. Which one? DNA, RNA, or both? The answer should be RNA. No oxygen is present in the sugar. That would be DNA. Deoxy, D is a root word, and is a nucleic acid. Both are nucleic acids. So you need to know the 3 types of RNA. And so there is mRNA, which is the messenger that sends a copy of DNA essentially to an organelle called the ribosome. Then there's rRNA, which we really didn't talk about at all. So just know that there is an rRNA and it's in the ribosome to help make the protein, but that's all it does. And then there's tRNA, which is also very important because in tRNA, it has the amino acid that is needed to begin to make the protein, which proteins are a bunch of amino acids put together. So tRNA and mRNA are kind of the most important of the RNAs that we're going to talk about. But just know that mRNA or rRNA exists. All right, so protein synthesis. Protein synthesis has 2 processes to make a protein, okay? And so let me give you the flow chart. So we start with DNA, and DNA is so precious. It's so important and valuable to cells that it cannot leave the nucleus. So that's why we take and we make a copy of it, or we transcribe it, we rewrite it like a recipe to send out of the nucleus. And so we take DNA molecule and we go through a process called transcription to make mRNA. Then that mRNA is going to leave the nucleus and go to a ribosome to go through a process called translation. And then that will help make our final product, which is the protein. So transcription makes mRNA and translation makes proteins. And proteins are really important because they're just determine the traits and their enzymes and their channels in our cell membrane, and proteins just do so much. And so we need proteins. And so protein synthesis happens all the time in our cells. So looking at this picture, would this be transcription or translation? Just looking at the picture. Based off this picture, this is transcription. Now, how do we know? Well, this big green thing is called RNA polymerase. We are inside the nucleus because here is DNA, okay? So we know that transcription happens in the nucleus. So we're in transcription. So we have RNA polymerase, which is an enzyme that helps this process happen. And you can see that DNA is opening, and this right here is our little recipe from DNA. It is our mRNA. It's our messenger RNA, our mRNA right here. And you can tell that's because it's single-stranded. It has U's on it, right? So this is transcription. So that's kind of all you need to know about that. Taking a look at this picture is the process of translation. Now, how do you know it's translation? Well, there's no DNA, so we got to be outside the nucleus. We see this right here. This is just a single-stranded long piece of nucleotide, so that must mean that's the mRNA. And here's the U's to tell you it's RNA. And then we're in this structure here, this big thing here, and that is a ribosome. And remember, when we talked about organelles, ribosomes make proteins. They're the first process of building a protein. So we're in a ribosome. Now these are tRNAs. So some things to remember is that we in the ribosome reads every 3 letters. Every 3 letters. And we call these 3 letters codons, right? So AUG is 1 codon, UUC is 1 codon, AAU is 1 codon. And so tRNA has what we call the anti-codon, which only matters when we're talking about how they match for a second, so they bring in the amino acid it's needing. So you really want to use mRNA when you're trying to determine the amino acid that's coming in, specifically with the genetic code chart, which we'll practice in just a second. But just remember that tRNA has an anti-codon which matches the base pairs, only so that it helps bring in the amino acid. So a tRNA comes in, brings the amino acid, leaves.